Good morning, students. I hope you're all well, staying warm. It's right now. I'm recording this on Monday, and it's I don't know, 18 degrees outside with a lovely, beautiful white covering of snow everywhere. Beautiful, but still very, very cold, and the roads are very unsafe. So we have a lecture which will be easy to do. As you go through this, you can pause it whenever you like if I'm going too fast, or rewatch something if you like, if you didn't get it the first time. The only thing that concerns me is oftentimes you have questions as we're going through this. So be sure to have a little pad of paper or something next to you and write those questions down as you think of them. And then you can either email me with that question if you like, or you can wait till next week during our lab time and I can answer some of those questions for you. All right, so we're looking at the circulatory system, which is very, very fascinating. Of course, you know I say that every week, but we're going to look at blood, and then we're going to look at the system itself, and then we'll finish with talking about the heart and the veins and the arteries. And so next week when we get together, we'll have a heart dissection and do a couple other things. We'll be checking blood pressure, stuff like that. All right, so the circulatory system, let's get an overview of it. It involves several things. It involves our blood. Hemopoiesis, remember that? Hemopoiesis is the manufacture of the blood. Remember where that happens? Can I hear you out there saying it? In the bones, that's right. So in the red marrow of the long bones. Hemostasis is another word that sounds like that, but hemostasis, not to be confused with homeostasis. Hemostasis is actually the way the blood can clot to keep us from bleeding too much. We'll be talking about blood typing. Typing would tell what type blood do you have, and the types of blood that humans have. We'll be looking at circulation, how it goes through the system. I want you to think about the circulatory system. Instead of being a circle, I want you to think of it like a figure eight, okay? So we have our heart in the middle, and it's the pump, and it's pumping down to the body and back to the heart and up to the head and back to the heart and down to the body. So sort of like that figure eight situation going on there. We'll look at the heart itself. It's a beautifully designed pump. And the cardiac cycle, how it actually pumps, you know, how the muscles contract. It's a special kind of muscle tissue called cardiac tissue. And then we'll see when the heart does push the blood out, it causes the blood to kind of slam up against our blood vessels. We have different types of blood vessels, arteries, veins, and capillaries. We'll see the differences of those. But the blood pressure is talking about when it slams up against them. How does the vessel react? How does the artery react? And then there's, of course, a condition called high blood pressure. We'll look at that as well. Okay, so let's start with blood. I think this is where you're starting on your writing down your notes. It says Lab 22 at the top. My page number is 65. I don't know what yours will be, and of course we know, Gabby, you have no page numbers. Sorry again. Okay, so but at the top of the page we have blood functions. So here we go. This is going in those boxes. First of all, transport. Okay, blood is responsible for transporting things to the tissues. Basically, it's transporting nutrients and gases. So the gas the tissue needs is oxygen, and then of course CO2 is the waste product from our metabolism. That's going to be transported as well. It's just transported to go out of the body. It's going to go to the lungs where we can exhale it. We'll be taking nutrients from our food sources, taking that to the tissues, and then wastes from our tissues out of the body once again through the kidneys. And then, of course, we know about hormone transport goes through the blood. 2, we're looking at regulation. Okay, it's the blood that decides, you know, if we need some changes to take place. For instance, our fluid volume. Sometimes we don't want our volume of our blood to get too thick. That can happen if you're dehydrated. And other maybe medications can cause that kind of thing, and so our body will try to respond by so what happens when your blood's thicker, it causes it to have more sluggishness. It also can increase the blood pressure because our heart has to try harder to push that sluggish blood around. Also, if there's a temperature situation, you know that if you go outside and you get all hot, how you turn kind of red, that's because your body's pumping blood to the surface of your skin to cool you down. In addition to, of course, we know sweating, right? pH levels, if those should change widely for some weird reason, then our blood is going to know it and go take action to correct that. Protection. All right, so our blood's going to protect us against fluid loss. We do that with blood clotting. The blood will clot to keep us from losing that blood. As it may be easily understood, losing a lot of blood is bad for you. Also, we have white blood cells that get distributed in case we have some sort of infection. So you see that WBC. WBC stands for white blood cell, and we'll see the RBC as well. Okay, so you've got those three things in your three boxes. Meanwhile, some other characteristics. These are some definitions you'll be writing down. Viscosity is the resistance to flow and the alteration of shape due to cohesion. Okay, so we'll be looking at the thickness of blood here. All right, so if the blood is really thick, it'll be harder to push. Think about it. Think about syrup on a cold day. If it's outside, it doesn't flow very well, but if you warm the syrup up, it flows really nicely. Also think about the fact that blood has a lot of water component to it, and water does stick to itself. That's what cohesion means. And so if it gets thicker, it's going to be kind of clumpy, if you will. Although I don't like that word because it sounds like it's clotted. That's different from clotting. It's just viscous. It's thick. All right, plasma is the fluid portion of our blood. It's mostly made up of water. I'll show you a picture in a minute there. Formed elements are the solid portion of your blood. So we have the blood cells. Erythrocytes are called red blood cells. Notice that's RBC. RBC stands for red blood cell. And then we have our leukocytes, are our white blood cells, WBC. And then we have these little things called platelets. They're fragments of cells, and they aid in clotting. All right, so we'll see the whole very complex process of clotting here in a few minutes. All right, so here are some drawn pictures of what these look like. They are not to scale because the thrombocyte, which is the platelet on the right there, it's very, very small. Because remember, it's just a fragment of a cell. It doesn't have all the regular things a cell would have, for instance, nucleus and all of those organelles and this kind of thing. They are just designed to get really sticky at a certain time when they're told to get sticky. And when they get sticky, they stick to each other, causing a clot. Our leukocyte, which should be larger because it's the biggest of the cell. A leukocyte is a white blood cell. And then our erythrocyte, you see right there, it's an interesting cell as well. It looks a little bit like we're going to call it a donut, but with the hole, not a hole in it, but a depression in the middle of it. Kind of like a thumbprint cookie kind of a thing. And that's because red blood cells do not have a nucleus. Well, that's weird, right? Because how in the world do you make new red blood cells? Well, they're made in the bone. So all of the red blood cells are made in your bones. And therefore, these red blood cells don't go through mitosis. Do you get it? Yeah. So they do their job, which is basically carrying oxygen to the tissues, carrying CO2 back to the lungs, and then they're done. They only have a lifespan of about 4 months. And because of that, our bones are constantly making new blood cells, and the old ones go to the liver to die, to be reestablished there, to take them apart basically. So if you look at blood, so when I was in the clinic, oftentimes I would get a blood sample. It would come in different types of test tubes. A red top tube would be considered a clot tube. If you've ever given blood before, you probably have noticed they put it in a few different kinds of tubes. One of the tubes is kind of large with a red top, and the other tubes are smaller with purple tops. Well, the purple top tubes have an anticoagulant in it. It keeps the blood from clotting. As soon as your blood comes out of your body, it's going to want to clot because it has contact with the air. And so when you have a clot tube, you want it to clot so that you can then take that tube. You can see on the picture here to your left, that tube has been spun down in a centrifuge. So that's what I would do, is I would take these clot tubes, put them in a centrifuge, spin them down. All of the solid portions would go to the bottom of the tube. And you can see that would be red blood cells. And then there's a little thin area in the middle where you can see a little white stripe, and those would be the white blood cells, and some platelets in there as well. And then the top portion is the liquid portion of the blood. That's called plasma. Some people call it serum. Most people say that it's straw colored or yellowish colored. All right, so blood is viscous. That means it's pretty thick. It's dense. That means it's heavy. One of the reasons is because there's iron in it. I'll show you how in a minute. It has a pH, a very narrow pH from 7.35 to 7.45. It doesn't want to be out of that area much at all. a volume of about 5 liters in an adult, probably adult male. 55% of it is considered plasma. So the upper part of the tube there, 55%. Of that plasma, 91% of it is water, 9% is made up of proteins, blood gases including O2, CO2, ions, and waste products at any given time. The proteins include albumin, which help regulate water, globulin and fibrinogen, which are there for blood clotting. So I think you're going to be writing a few of these things down on these blanks that you have right here. Okay, 45% of the blood includes those formed elements. 99% of those formed elements are RBCs, erythrocytes, and 1% are WBCs and platelets. Okay, so the vast majority are indeed the red blood cells. There's lots of those, comparatively speaking. Okay, so I think I want you to write down on your blanks under plasma. You wrote a couple of things. Go ahead and write that 91% of it's water, and then say proteins. I'd like you to go ahead and write down those proteins: albumin, globulin, and fibrinogen. And then under your erythrocytes blank, go ahead and say 99% of the formed elements are RBCs. Okay, does that work? All right, let's look a little closer at those RBCs. On your book, you see there's going to be a box at the bottom of the page saying white and red blood cell facts. We're going to do that first, then we'll go back up to figure 11.3, which is actually showing you what one of the components of a red cell looks like. And you're supposed to draw it. We'll see about that. Okay, red blood cells have a unique shape. Like I said before, they are anucleate. That's the word for saying it does not have a nucleus. So that kind of donut shape, I kind of like to think of them if you're going down, if you're tubing on a river, you know those big round tubes that have the mesh in where you sit? You know what I'm talking about? That's kind of like what it reminds me of a red blood cell. It's got that thin portion in the middle right there. They don't have any DNA because they don't have a nucleus. They do not go through mitosis. They have a short lifespan. Their main job is to carry gases, that would be oxygen, on a molecule, on a big protein called hemoglobin. All right, hemoglobin. That's what you're going to draw in that box here in just a few minutes, or you're going to try. Their red blood cells are continuously made in the bone marrow. Oh, look at there, 2 million per second. If you have a low number of red blood cells, or your hemoglobin, a low level of hemoglobin, that's called anemia. And we measure that with a test called an H&H, a hematocrit and a hemoglobin test. Essentially, you're just putting blood into a little tiny glass tube that gets spun down in another specialized centrifuge, and you can see how much red blood cell you have there, and it'll give you an idea if you're anemic. Sometimes your color can tell you a little bit about that, especially with animals. We would have little kittens coming into the clinic, and if you lift their lip up, you know we have pink gums, yes. So if you think about this, if you have a dog or cat, you can try this. You push your finger onto their gums, and you let go, and it turns kind of white because you squeeze the blood out, and then you let go and it refills with blood. So we'd have these kittens coming in that had fleas all over them, so a flea infestation, and fleas suck the blood from an animal, and they would have no refill. So in other words, their gums were not pink, and when we pushed on them, they were kind of white looking, and we pushed on them and there was no refill right there. They were anemic, and that was trouble. So the first thing, of course, was to get the fleas off of them, and secondly, we would try to rehabilitate them, even to the point of sometime giving them a blood transfusion, which is a pretty serious deal. So you want to keep the right number of RBCs and not have anemia. In humans, anemia can be caused by several things, bleeding for one, and then possibly some diseases. And even some drugs, some medications can cause some people to get anemic. An H&H for males is between 44 and 48, and for females between 38 and 42. In other words, men have a little more red blood cell content. Sickle cell anemia is an interesting genetic deformation of the red blood cells. Okay, so red blood cells are supposed to look round like this. See that kind of donut look I'm talking about right there? And that's they're going to be carrying that oxygen on the hemoglobin there. But sickle cells look like that, so they're not round. And the reason that is, it has to do with the shape of the hemoglobin itself. All right, so here we have regular normal red blood cells going through the vessel. And see how the way they're shaped, like that raft I was saying about, they can be squeezed and moved into the position they need to be. And as they're pressing up again, you can see my pointer. Oh, good. Oh, no, you can't. All right, so those red blood cells going by themselves, single file, do you see how they're being squeezed up? When that happens, look, they're touching the side of the vessel, and they're able to exchange those gases very easily. And that's why they're shaped that way. And you can see on the small picture down below that the normal hemoglobin is inside that red blood cell floating around in there. Now on sickle cell, look at the bottom right picture. The hemoglobin is all stretched out. It's almost like it's been stretched into a long stick versus the coiled nature of it that I'm going to show you in just a minute. And because of that, it causes those blood cells to be shaped in that sickle shape or that curved shape. And they're stiff. So notice how they're getting caught and clogged up in those little vessels. So this does not allow the flow to take place. So who has sickle cell anemia? Well, it is a genetic disorder. The DNA codes for it. It happens to a child. So you don't have it. You would know it by now. You would have had serious complications from this. I will say that it's much more common in African American people, and so that has to be checked when they're very, very young. I'm not sure, to be honest with you, what we do for it, because DNA is causing our blood cells to be made that way. So I'm not sure if there's something we can do to help thin the blood or something like that. But the hemoglobin is obviously not carrying it very well either. Now this is what hemoglobin is supposed to look like. So think about, these are all folded up on one another. They're proteins. They're 4 proteins that are put together, folded up. And inside you can see that little rectangular thing with the round deal. That's called a heme. And it's called that because it has an iron molecule on it. So heme has to do with iron. And that iron, guess what it holds? It holds an oxygen. So each one of those hemes in there will hold an O2 molecule, and that will be inside the red blood cell being taken to the tissues. So you may want to pause the video right now and see if you can draw this in your figure 11.3, I believe is what you're supposed to draw there. Good luck with that. If you want to take a picture or something like that instead, that'd be okay. All right, now moving to white blood cells. So now we're back down in that lower box where you wrote some things about red blood cell facts. Now write some things about white blood cell facts. All right, leukocytes exhibit something called amoeboid movement. It's called diapedesis, and I believe on the next page at the top you will see that word up there. So you can write down for diapedesis, it means the way that the cells move to squeeze out of the blood vessel. Now, why do they squeeze out of the blood vessel? To get to the tissues that need white blood cells. What tissues need that? Tissues that might be infected with something. What is the white blood cell going to do? It's going to eradicate that thing. It's going to eat up that invading foreign thing. You know, it's a bacteria or something like that that has come in. Chemotaxis is another way to talk about the way it's moving. Okay, so the attraction of the cells to a chemical stimuli. If a stimulus of a bacterial infection is given, then a signal is sent out by way of chemicals to bring white blood cells to the area. So it's going to be white blood cells coming to the rescue to go and take care of whatever has been infected. Which brings us to phagocytosis. We know this word. Cell eating. It's when the white blood cell will engulf and digest foreign cells or even dead cells that need to be got rid of. When a lot of white blood cells end up in a place and they engulf a lot of stuff, they end up dying, and those old nasty white blood cells are known as pus. So if you've ever had an infection that had some pus associated with it, those are just your white blood cells who had come to do their job. So it's a good thing, and that's the leftovers, if you will. So there are specialized types of white blood cells. So they're not all the same. Some are called granulocytes. Don't write that down just yet. And they stain in such a way that show large granules inside of their cells. Others don't have those grains in them, so we call those agranulocytes. And that's just, like I said, the way they stain. We will be staining your blood and doing a blood typing for you, but not until our next module, okay? Because we'll be looking at that as far as surface antigens on there. And when we do that, we'll take a look at your white blood cells then. Okay. So here is a drawing of the different kinds of white blood cells. You can see at the top right there are the 5 main types of white blood cells. A monocyte, a lymphocyte, a neutrophil, or called a segmented neutrophil, an eosinophil, my favorite, and the basophils. And then you can see our erythrocytes and platelets down there. The macrophage, that picture, I believe it's referring to the beginning of a white cell and when it's not differentiated to become one of the other types of white cells. So just to call the macrophage, because sometimes the monocytes are also considered macrophages. They're the big eaters. They can engulf a lot of bacteria. So here's what I want you to write down, however. The white cells right here. In your blank called granulocytes, you're going to list those ones on the right there. Okay, so we have an eosinophil. We'll talk about each one of these individually in just a minute. A basophil and a neutrophil. Neutrophils, or segmented neutrophils, are called that because of their nucleus, how segmented it is. Those are by far the most commonly found. So those are the granulocytes. Put those in your 3 boxes. And the agranulocytes are those other 2, okay? The monocyte, which is the big one, the big eater, and then the lymphocyte, which is very specialized. We'll be talking a lot about lymphocytes when we look at the lymphatic system in next module, and which is why, once again, we'll be looking at our white cells then instead of next week. Okay, so let's start with our segmented neutrophils. The most common white blood cell out there. Look at it, looks kind of like a smiley face. If you turn it over, it looks kind of like headphones or something like this. So my job at my clinic, once again, was to take the blood and make something called a diff slide. Diff, D-I-F-F, stands for differential. And I would be able to go in and look at the different white cells and count them. So right there, that picture you're looking at shows one big white cell in the middle. Those smaller cells around it, those are the red cells. See how thin their middle parts are? You see through some of them, in fact. And that little chunk right there, probably at about 1 o'clock, if you're talking about a clock, that's a platelet. So first of all, you can see how much smaller the platelets are, and furthermore, that the white cells are larger than the red blood cells. All right, so what is the job of a segmented neutrophil? It's to gobble up bacteria that might have invaded. They can gobble up about 10 bacteria, then they die, and then they become pus, like we said before. All right, when I'm doing a diff slide, I count 100 different white cells. Okay, that way my math is easy, right? So I can tell a percentage very easily. So typically on a diff count, I'm going to be looking at somewhere in the neighborhood of 80% of them being the segmented neutrophils. Okay, a basophil, they're large, they're kind of rare. Remember, they're granulocytes, so we can see those grains in there. They release something called histamine, which cause inflammation. Okay, inflammation can be good in that particular case, because what it will do is it will bring other cells to it. It also releases something called heparin. Heparin is an anticoagulant. It keeps blood from clotting. In this particular case, it would slow clotting down. Once again, a good thing. We're bringing all those cells to a place where there's some sort of need. It speeds up healing by bringing lots of oxygen from the blood, and immunity factors, namely lymphocytes, which is a different kind of white cell we'll see here in just a minute. Eosinophils, the reason I like these is because while these granulocytes say that they stain those grains in there, really you can only see it on the eosins. All right, so an eosinophil has massive, giant granules inside there, and they're really cool to look at. Sadly, they're kind of rare. So oftentimes when I have students look at their diff slide of their own blood, we don't find any eosinophils. There's very few of them. They will be increased, though, in particular conditions. For instance, if you have an infection of a parasite, a parasitic infection, eosins will increase. So they show up very, very commonly with parasitic infections, which, of course, with dogs and cats, you see, I saw them more frequently because dogs and cats have lots of parasites, intestinal worms, heartworms, other things out there. Humans can get it too, but mostly in kind of tropical locations. I have seen it though on a student, and if we find anybody with one of these when we do that in our next module, we'll make sure everybody gets to see it. All right, lymphocytes are one of our agranulocytes. They're the smallest of the white blood cells. You can see it there next to some red blood cells. It looks a little bit like a fried egg with not much white part, right? They're the second most common. So I would say if I had 80% neutrophils, I probably would have about, 12% lymphocytes, something like that, unless there's an infection. If there's an infection, the lymphocytes will go way up because lymphocytes are really the big fighters when it comes to secondary immunity and things like this, which we will talk about in the next module. So you can see right here that all of our, one thing that's cool about lymphocytes is that not only are they made in the blood, They go camp out inside your lymph nodes, and if they're needed, they will proliferate inside the lymph nodes. In other words, they can be made elsewhere besides the bone. You get it? They can cause mitosis right there. They can have a quick proliferation inside the lymph node in response to some sort of infection, including even viral infections. You can see on our picture to the right here, it's showing a bunch of stuff right here talking about antibodies and plasma cells and our different types of immunity, cellular immunity and humoral immunity. We'll talk about all of that in Module 12. Fascinating stuff. We'll get to the details. It's a little bit more difficult stuff to understand. That's why it has its own module. Okay, our monocytes, look how big they are. Have you heard of a disease called mononucleosis? That is when you have an overproduction of monocytes. All right, so here they are. They're huge. Typically, mononucleosis is caused by a viral infection, and it causes you to be very tired, and they call it the kissing disease because it spreads from one person to another fairly easily. But you don't have to have been kissing somebody to get mononucleosis. You can get it from other places because viruses just tend to travel. But if you do a blood count and you see lots and lots of monocytes, then you can guess that the patient may have mononucleosis and then treat them for a viral infection. Okay, so you know about hemopoiesis. That is the manufacture of blood. The formed elements are made in the red marrow of the long bones. And our stem cells differentiate into the different type of blood cell they want that the body needs. The vast majority of them, you'll see to the left, are going to be made into red blood cells because we know that, what did we say, 90% of those are red blood cells, so we need lots, or 99, right? So we need lots of those. So notice our stem cell in our bone, though, is going to decide what's needed. If I need red blood cells, I'll make erythroblasts, making erythrocytes. If I need some big white cells, we see our mega karyocyte right there. It's going to turn into platelets. It's going to fall apart into platelets. And then look at our myoblast turns into the different kinds of granulocytes. And then we can see our other ones on the other side. Notice our lymph node kind of comes from a special stem cell there. And versus also, see the natural killer right there? We're going to talk about that next module. That is where it proliferates in the lymph node. Okay, so it's a little different that way. Pretty fascinating stuff right here. All right, so making blood sounds simple, but it's not simple at all, right? The red cells may be pretty easy, but all this other differentiation is pretty fascinating stuff. Okay, so hemostasis is the process by which the body stops blood loss. That's blood clotting. Okay, so there are 3 stages of this. You have this at the bottom of one of your pages right here. So what is hemostasis? The process by which the body stops blood loss, or blood clotting to be simple. The 3 stages are these. Number 1, vasoconstriction. That means the constriction of the veins. Okay, the smooth muscle of the vein closes up at the breach. So to say you've had a cut, that's going to have that jagged edge. It's going to close up. I will say jagged cuts heal faster versus if you cut yourself with one of those scalpel blades, really, you know, it is a very clean cut. It actually bleeds a little bit longer. You may have noticed that before. But the first thing to happen is the constriction of the vessel itself. Next, the platelet plug stage. So now platelets will come to the area. They swell up. They get sticky. Why does that happen? Typically it happens because they've been exposed to oxygen. But other chemicals in the body will say time to get sticky. For instance, if you have an internal bleed or something like that. Some sort of trauma usually exposes collagen fibers to air or whatever, and that triggers the release of the thromboxane to a cut vessel, and platelets will gather there. The clot forms. It's called a thrombus. You'll get that here in just a minute. Sometimes if the clot breaks loose, it's called rogue. It runs through the body. That's very, very bad. Sometimes it'll clog up inside a rough vessel and let this rogue clot go through your body, which can end up causing a heart attack or a stroke if it ends up in your heart or ends up in your brain. If one of those clots breaks loose, it can travel to a small vessel, whether it's in your lungs, in your heart, in your brain, even in your legs, you know, some small vessels anywhere, causing what's known as an embolus. And then our 3rd stage is called the coagulation stage. This is where you're going to see it gets very, very complex, where we have a cascade of biochemical events. Yikes, right? We know to be afraid of that. So this, I wanted to show you this picture right here. Here's our two words. Thrombus is a platelet plug designed to stop bleeding, right? It's a good thing. But sometimes when it goes rogue, so you can see right here, that's called an embolus. Look on the left picture right there, you can see a rough interior of a vein where we can sometimes have this collection of various cells, including platelets, including some old red cells, some young red cells, will kind of clog up there. And if one breaks loose, it can travel to your heart, to your lung. That's called a pulmonary embolus. That has dropped full-grown men, killed them immediately. Also, it can go into if the clot ends up in the vessel feeding the actual heart, that causes a heart attack. If it ends up in the brain, that causes a stroke. And then you can have them, like I said, in your legs, causing what they call deep vein thrombosis. Bad. We don't want that, right? Which is why it gets very complicated to clot our blood. Okay, so clotting the blood has to go through a cascade of biochemical events, and you can see a picture of that right here. Okay, so you can see the word that says trauma. We're looking on the right side here, tissue factor, what's known as the extrinsic pathway. You can see trauma takes place, and we have these things in our blood, these proteins in our blood, that are called factors. Okay, the factors used to all have these cool names like Christmas factor and such, named after people and this kind of thing. But now we have Roman numerals, which I can't stand Roman numerals, but that's okay. I want you to look on both sides. We have the extrinsic pathway and the intrinsic pathway, which has a damaged surface, and you can see what happens. Okay, so let's start over there. Look on the left. We have tissue factor or blood clotting factor 12, and when there's a damaged surface, that factor 12 will be converted to factor 12A, and 12A will cause factor 11 to go to 11A, and 11A will go over to X, so it's 10 really. So notice X on both sides. So each factor pathway is trying to get to factor 10, otherwise known as X. Okay, so we see our X over there? All right. So when X is activated by 11A on the left side or 7A on the right side, then we have an activated factor 10. And what that causes is it causes prothrombin, which is already in your blood, to turn into something called thrombin. Thrombin also has a factor called 2A. So thrombin will then influence fibrinogen, which is factor 1, already just in your blood, flowing around, but it'll cause it to turn to fibrin, which is 1A. And then 1A, you see we have then 13A right there. We have cross-linked, this kind of thing. But when we get over to fibrin, then we're going to see that fibrin is what is causing the clot. Okay, so maybe this picture will be easier to understand. Oops, sorry. Okay. Can you see this? Can you see my pointer now? You can. Okay, here we go. So notice here's 10, remember X right there. So we're all trying to get to 10 by taking 12 to 11. Remember those A's are in there. This is simplified a little bit. To 9 to 10. And over here on this other side from 7 to 10, which then takes 5 or 8 and turns thrombin, which is already in the blood, and it's going to turn it into fibrin. So fibrin is down here in this blue box right here. And we see 13, factor 13 is influencing that as well. So our platelets are now activated, our fibrinopeptides are activated, and that results in a clot. Okay? So we have various things that influence it. We have something called tissue factor, we have something called von Willebrand factor, and then other precursors and things that take place. So you see how this cascade of events, the whole purpose for this is that we don't get a clot unless we need a clot. Okay? Because you don't want those rogue clots going in and causing your, strokes and your heart attacks and your emboli and this kind of stuff. You don't want that. So you do want a complicated process to get this. I will tell you that there are scientists out here who have studied this complex process, this cascade of biochemical events, and it has caused them to become believers in God, because there's no way that it could have taken millions of years between each one of these steps. We would have bled to death. Okay, so this is one of those beautiful examples of God's incredible design. It is fascinating what he's done for us here. Thank you, God. This is what a clot ends up looking like. So you see the red blood cells, you see that netting around there? That's the fibrin. So the fibrin will hold on like a net around those blood cells, and the blood cells can't move anymore. They're trapped in a net. And that is a clot right there. Pretty cool, huh? All right. We're going to watch a little video about it.
The process of hemostasis has many components and interacting mechanisms that often appear complicated and difficult to grasp. Multiple factors activate each other from different pathways under the control of various enzymatic reactions in a very convoluted manner. However, if looked at from the perspective of what happens at the injury site to specifically stop bleeding, it is in fact quite simple to understand. Only two blood components emerge as necessary: platelets and fibrin. The following animation will highlight the process by which they work together to achieve hemostasis. The endothelial layer in healthy individuals serves to protect against spontaneous accumulation of platelets and fibrin in the vessel wall. In the event of vessel wall injury, bleeding will occur. Hemostatic plug formation is initiated upon exposure of the subendothelium at the injury site. Vasoconstriction in the region of injury results from the release of thromboxane A2, a major enzymatic product of platelet activation which causes vessel wall constriction. Von Willebrand factor is secreted by the endothelial cells and binds to exposed collagen fibers at the wound surface. Platelets then adhere to the wound through their interaction with bound von Willebrand factor. This process constitutes platelet adhesion. Adherent platelets are activated and attract other platelets through a process known as platelet aggregation. Fibrinogen molecules form bridges between adjacent platelets. This forms an aggregate of platelets that occludes the wound and eventually stops bleeding. This unstable primary plug of loosely aggregated platelets must then be consolidated into a more stable plug. This is achieved through the generation of thrombin at the wound site. Free tissue factor released from subendothelial stores and platelets initiates the coagulation cascade with its interaction with activated factor 7. The complex of tissue factor and factor 7A converts factor 10 to its active form, factor 10A. Tissue factor 7A also converts factor 9 to its activated form, factor 9A. However, further generation of factor 10A is inhibited by the tissue factor pathway inhibitor. At this point, the amount of factor 10A produced is insufficient to sustain coagulation. Further factor 10A, to allow hemostasis to progress to completion, can now only be generated by the factor 9A pathway. By this stage, though, enough thrombin has been generated by the factor 10A to activate factors 8 and 5, which both act as potent catalysts. Factor 8A increases the capacity of factor 9A to activate factor 10 to 10A many thousand times. The increased factor 10A produced in this way, along with its own cofactor activated 5A, forms a complex that promotes the efficient conversion of prothrombin to thrombin. It is thrombin that then allows the conversion of fibrinogen to fibrin. While the process of the coagulation cascade as outlined can be confusing and difficult to retain, it is most important to remember the importance of the final result, fibrin, in wound healing. Fibrin is the biological glue that eventually seals the hemostatic plug and ensures hemostasis. As soon as a small amount of thrombin is formed, the clotting process accelerates and provides more and more thrombin into the wound. At high concentrations, this thrombin quickly converts fibrinogen to fibrin on the surface of the platelet aggregate to stabilize the hemostatic plug. Over the course of the subsequent 7 to 12 days, the process of fibrinolysis dissolves the fibrin in the wound as the site of injury heals, and the cell layer in the vessel wall is restored. Some scar formation occurs, and within a few weeks, the wound is completely healed.
All right, so did that help explain it to you a little better? It's very complex, obviously. Okay, so we're going to go through in your book right here. You had your 3 stages of hemostasis. Now you have your 3 steps of coagulation. All right, so that 3rd stage, that coagulation stage, that's this fancy one we've been talking about, okay? So that's where all these factors have to be activated. All right, so let's go through there. First of all, the thing we have is trauma. Something happens where the blood vessel has been disrupted. Either it's been cut or damaged, even a bruise, something like that can cause that trauma. So when trauma happens, then we have prothrombinase, which is already there, but it is now activated so that it can produce thrombin. Now, it's easy for me to say that, right? But you see all those factors that had to trip before I got to factor 10 and 10A, bringing in 5A to help to get this thrombin going. And furthermore, it's not going to continue, that cascade's not going to continue unless there have been lots of signals saying we need more clotting here, because we do not want to make a clot accidentally. We don't want one to be floating around without a place to be, closing up, right? We don't want it rogue. All right, so our thrombin is made, which now will trip some more of our factors, and our fibrinogen, which is already in the blood, will then be influenced to produce the fibrin. And the fibrin is the net. So what you're putting in your 3 steps of coagulation is thrombin, fibrinogen, and fibrin, okay? So thrombin, fibrinogen, and fibrin. All of this, by the way, is assisted by something called vitamin K. Vitamin K, there's certain types of vitamin K, 1, 2, 3, that kind of thing. And one of them, I believe it's K1, which helps with the clotting. It is made in your gut by bacteria that live there. Okay, so these bacteria that are living symbiotically with us are actually manufacturing vitamin K. It turns out that vitamin K is going to help with the blood clotting, and if you don't have it, you won't clot as well. Okay, so the vitamin K enhances that, enhances the ability for the factors to be activated. And so it takes about, once a baby is born, remember, it's being made by bacteria in the gut. When a baby's born, they don't have any bacteria in their gut because they've been in mom, right, the whole time. They've been getting their nutrients through the placenta, not through their digestive system. And so it takes a few days. It takes, in fact, 8 days for the baby to be exposed to the bacteria in a normal condition while breastfeeding and other contact through, living in the world. That will cause the vitamin K to be produced by the bacteria that start to grow inside the gut. And that is why, you may not have thought about it, when they do, little boys, they do a circumcision. In the Bible, it talks about that, involves cutting, and that cutting needs to have clot abilities. So in the Old Testament, it said that baby boys would be circumcised on the 8th day. And the reason it's the 8th day is because by then, the vitamin K is produced by the body. Pretty cool. So there you go. And we, if you have any questions about that, we can certainly talk about that and next week. Okay. Here is just a little GIF about the blood nicely flowing through the vessel right there. Everything's nice and smooth, so it's flowing through beautifully. We're part of the plasma, I guess. And what happens if the flow slows down? Okay, what will happen is if we stop flowing, we do have coagulation. So think about this. When somebody dies and their heart stops beating, they don't have any flow. What happens naturally to the blood? It clots. Okay, so it's interesting to think that flow has something to do with it. We also know if it's exposed to oxygen, the clotting process will begin. All right, let's talk now about blood types. Blood types, there are 4 different blood types. The blood type is determined by surface antigens on the red blood cell. Okay, so these are on your RBCs. All right, it's determined genetically from your parents, and there are these things called surface antigens. So you can call them antigens if you like. I like to refer to them as surface antigens so you can remember they're on the surface. They're on the surface of the red blood cell. Okay, because there's another word we'll be talking about in the next module called antibody, and it's kind of easy to get those two mixed up, an antigen versus an antibody. The antigen's on the surface, so if we call it a surface antigen, that'll remind us of that. The surface antigen will trigger production of an antibody. An antibody is the body will make something to fight a surface antigen of an invader. Okay, so that's part of our lymphatic system, that's part of our immune system. So that's another reason we will be testing your blood type, but not for this module, for the next module, because we'll be looking at antibodies, and therefore the antibodies will test your red blood cells to see what antibodies you have on there. And I'm really, really hoping that some of you have some type that's not O. I have type O. That's the most common out there, but the problem with O is it has no surface antigens in respect to blood type. Therefore, my antigen antibody test is really boring. But if some of you are A or B or AB, I hope one of you is AB, we can see some really good response to our antibody testing for your blood type. There's another surface antigen also called Rh factor, which we'll get to that in just a minute. It's just another surface antigen, and it just happens that the majority of people have it, and that's called Rh positive. Okay, so here are the different kinds of blood types, okay? So we have our red blood cells, there they are on the top, and we have type, what are we looking at? Type A is that first one. So notice its surface antigen has that little round shape on it, okay? Our type B, notice it has a different surface antigen. Now these, of course, have been oversimplified to help us to understand, okay? Now look at type AB. This is a case of incomplete dominance right here. So A and B are not dominant one over the other. They're co-dominant. Okay, so they have both then. Okay, so type AB will have both of those markers on their cells, both of those surface antigens, the roundish looking ones and the triangle looking ones. Look at type O. Nothing. There are no surface antigens with respect to blood type on there. Keep in mind, for instance, I'm O positive. I don't have any A or B antigens, but I do have an Rh factor antigen on there, so it's not completely naked. You know, there are other things on the surface of cells. We're talking in respect to blood type right now. Okay, now let's go back to type A over here. Notice on the surface we have those antigens, and see the next box down, it says anti-B antibodies. So that's an antibody. Look at the shape of it. It's like a Y shape. That will match up against that triangular shape on the type B cell. What that means is if I give type A blood to a person who is type B, the antibodies in type A will attack the cells that are type B. All right, you get it? So back in the time of when we were just figuring out about blood types, in war, when men lost lots and lots of blood, they would give them a blood transfusion, and they recognized that sometimes they were helped by it and their lives were saved. Other times they died more quickly. The reason was they didn't understand they were giving typed blood to somebody that was the wrong type. So if I give the wrong type of blood to somebody, it'll kill them faster. Okay? Now, we will talk about this again in Module 12. Is that right? Module 12. And so we'll be able to understand this a little better. Notice over on type O, I don't have any surface antigens, but look at the antibodies I have. I have both anti-A and anti-B antibodies in my blood. That means I'll reject everything. So the only blood that I can receive as a type O patient is type O. That's it. Okay? And we'll talk about how type O is actually the universal donor. I can give type O to somebody else because the blood itself doesn't have anything on its surface to attract any kind of rejection. It does have those antibodies in there, but they won't be a problem. And we'll discuss that next module. That might be a little confusing. Okay, so in your boxes here, you're going to write down some of the blood types. It's called a Punnett square. So let's say your dad had type A blood, but he was heterozygous for A and O. And mom had type B, but she was heterozygous for B and O. You remember that from from Biology 1, right? You can see we distribute these out here, and we can see the different blood types that are created right here. So some of the offspring will be type AB. We can see that in the top left box. Some of the offspring will be type O with B, but B is dominant to O. So therefore, they're going to be type B, okay? And then we see on the bottom square to the left, they'll have a gene for type A and a gene for type O, but A, once again, is dominant to O, so that patient or that offspring is type A. Now, the only way you can be type O is if you have two genes for O. Okay, because they're recessive to A and B. A and B are not recessive to each other, but they're dominant to O every time. So notice here I will have 25% chance of the children being type AB, 25% chance of the children being type B, 25% of the children being type A, and then 25% being O. Now you might wonder with this, with O being recessive, why the majority of the population is type O. Well, when I say majority, I say about 40%. Okay, so it is interesting, but that is the case. About 40% of the population is type O. All right, in your book you have several different ABO blood types. I think what you're supposed to do in that big box over there is to tell me the possible types of blood types there are. Okay, so there's AO, which would be A. There's BO, which would be B. There's AB, which would be AB. There's OO. So I'm not sure exactly how you're supposed to fill that whole thing out right there. Just as long as you realize that A and B have a codominance, and A and B are both dominant to O. Okay? All right. Universal blood type is O. We'll discuss that more because O doesn't have anything on its surface. It's not going to cause any trouble. But if you're going to go get a blood transfusion, the best kind of blood for you to get is exactly the kind you have. But in a pinch, if you're type A, I can give you type O as a transfusion. Okay? But if you're type O, you must have O every time. All right, what is Rh factor? It's just another surface antigen on red blood cells. It's common to about 85% of the population, so write that Rh factor. It gets its name because they studied it using rhesus monkeys. So that's where the Rh comes from. Now, what I want you to know about it, though, is it can present a problem in pregnancy, okay? So let's say there's a mom out there who has Rh negative, so they do not have the Rh factor, and their baby has Rh positive, okay, which they got that gene from the dad, right? Well, during that very first pregnancy, this only happens in the first pregnancy, everything's okay, the mom will generate antibodies against Rh factor. So then in the second and subsequent pregnancies after that, she will literally be making antibodies against her baby. All right, so this is a very difficult problem. Bad news. So what we need to know here, you are all girls, you're females, is do you have Rh negative, okay? And if you do, you just need to make sure when it comes time to have babies that you tell your doctor, I'm Rh negative. I had at least one student find out they were Rh negative in my class doing the test. So we'll find that out next module to see if you're Rh negative. Chances are you're Rh positive. It's 85%. So we'll see. Now, what happens with the second and subsequent pregnancies? Can they have other pregnancies? Yes. What they do is they treat the mother with something that discourages the body from making those antibodies. Not exactly sure the process, but they do treat it and everything's okay when they know. As long as they know, everything's okay. All right. We're now to 11B. We've covered kind of the physiology of things, talking about blood clotting and this kind of thing. Now we're going to look at the anatomy of the heart and the vessels. Okay, so go ahead and turn your page over to 11B. All right, here's our circulatory system right here. You can see on the right we have our heart and our lungs. So our heart is pumping blood to the lungs to get oxygenated, then back to the heart to pump it to our body to get that oxygenated blood to our body. It's known as a closed circulatory system. It's not open. There's no bleeding out into anything. Everything stays in vessels the whole time. Arteries carry blood away from the heart. Okay, so you see here in sketch 11.12, you're supposed to draw that thing right there to the right. Okay, so you can get working on that. But now I have things you're filling in below. Okay, so you see arteries. Remember, artery away. A for artery, A for away. Okay. Veins carry blood to the heart. All right. Capillaries are thin, tiny vessels where gas exchange takes place. That's where those red blood cells go in single file. It's so thin, they go through single file, and we saw how that allows for really, really efficient gas exchange right there. All right. Our next blank, pulmonary circulation. It goes from the heart to the lungs and back to the heart. Notice how close the heart is to the lungs. So it doesn't have to pump very far, does it? It just goes boom, boom, and then back to the heart right there. So that's the pulmonary circulation. The systemic circulation is from the heart to the body. So now, to get from my heart down to the tip of my toe, that's a lot farther. So that happens with actually the left side of the heart, which will then pump everything down to the rest of the body. So it turns out that the muscle on the left side of the heart is actually thicker. You can, because it has to pump farther. You can see right over here that thicker wall of the left side of the heart. You may be thinking I'm pointing to the right side, but it's left compared to where if I'm standing there with my body, it's on the left side. You get it? Yeah. All right. Now remember our figure eight? There it is. So there I'm pumping from the heart to the lungs, back to the heart, from the heart to the body, back to the heart, heart to the lungs. So there's my figure eight going on there. So here's what the heart looks like. It's made up of cardiac muscle tissue, which is a specialized kind of tissue. You are going to be writing this on the bottom of the page here. Interesting facts about the heart. Okay, cardiac muscle. It weighs about 10 ounces. It's about the size of your fist when you're an adult, I believe. It's a double pump sending blood through the cardiac cycle. So pump, 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 left, right, left, right. The heart rate is around 75 beats per minute. We'll check that next week. We'll take your pulse. It's nourished by the cardiac arteries. So you can see these arteries feeding the muscle of the heart itself, okay? These are called cardiac arteries, and they're very important. If those get a clot in them, that's called a myocardial infarction or heart attack. The whole heart is covered by a membrane called the pericardium. And we have large vessels going out of the top of the heart here. This one, the biggest artery in the body, is called the aorta. And notice it's going to curve downward here. That's taking blood to the lower extremities. But notice it's got three large vessels right here. Those are taking it to your neck and head, okay? Lots of blood to your brain, for obvious reasons. Then coming down from my head into my superior vena cava, that's our largest vein in the body. The vena cava, superior and inferior, just means it's below it, right here. And then we have our pulmonary artery. Notice it's an artery, okay, artery away. It has deoxygenated blood. Some people are tempted to say arteries carry oxygenated blood, veins carry deoxygenated blood. Well, that's not accurate, because right here our pulmonary artery is going away from the heart. But guess what? Where's it going? It's going to the lungs to get oxygenated, okay? So, therefore, it's an artery that it has deoxygenated blood in it. I just need you to understand that, okay? So artery away, vein toward. You can pause this and write those things down if you like. Okay, our cardiac tissue. This is still an interesting facts about the heart. It's made of its own class called cardiac muscle. It's involuntary. Thank you, Lord. We can't make our heart stop beating even if we try by thinking about it, right? By volunteering it. It's striated muscle, just like skeletal muscle is, but it has shorter cells. The cells are joined together at these things called I thought they were called intercalated disc. Okay, right here you can see that little intercalated disc right there. And they're highly excitable. In fact, they're so specialized that if you take some of the cells off the heart, they'll beat by themselves. They'll beat by themselves. Crazy stuff. Here's what it looks like under a microscope. You can see there's stripes in it. You can see those pretty well right here, the stripes, and then we can see one of those intercalated discs right there. All right, the internal anatomy of our heart. I believe you're supposed to be, okay, well, when we do our dissection, I don't guess you have to draw this, but just listen. It's got 4 chambers. If you know nothing else about the human heart, know that it has 4 chambers, please. The 2 sides are separated by the septum. It's a large area of tissue in there that actually contains a lot of electrical signals. The upper chambers are called the right and left atrium. They're atria, to be plural. The lower, larger chambers are called the right and left ventricles. The atria send blood to the ventricles. So it goes from the top to the bottom, just like a baseball game, okay? From the top to the bottom. And the ventricles send blood out of the heart. The left side goes to the body. The right side goes only to the lungs. The chambers are separated by valves. The left side has one called the mitral valve. I'll show you a picture in a minute. The right has something called a tricuspid valve. I'll show you that, and you'll see why it's called tricuspid here in a minute. Semilunar, that means half moon type of thing. Semilunar valves regulate blood flow to the aorta and to the pulmonary trunk. Okay, those are the ones going out, right? I want you to take your fists like this and put them together, okay? just like that. So you have your right fist and your left fist, and I want you to squeeze your right and then your left. So squeeze, 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 squeeze. I want you to think of the heart as a double pump. Pump, 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 pump. And your right fist, when you squeeze it, it's only going to the lungs. So it doesn't have to squeeze very hard, but the left one, boom, you have to really squeeze it hard because it's going to go to the rest of your body. Here it is pumping right here. So ready? Pump, 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 just like that. All right, so I like to think of that as our double pump. Here's what it looks like on the inside. Now, we color the heart red and blue, showing oxygenated and deoxygenated blood. I think one of you asked that question is, about blood being blue. Blood isn't really blue. You can see your veins in your hands, right? You can look at your hands right now and see some veins, and they look blue. It turns out that veins are blue, okay? They just happen to be blue. Now, deoxygenated blood is certainly not as red as oxygenated blood, that's for sure, okay? Oxygenated blood is bright red. You've seen it whenever you've bled, okay? You cut yourself, you see that bright red blood coming out. Well, it's just touched the oxygen in the air, and it's grabbing hold of that oxygen because hemoglobin is so incredibly efficient at grabbing hold of that oxygen, okay? Now, so what happens to deoxygenated blood? It's not as bright red. Let's call it magenta colored, okay? Maybe purplish, if you will. But it's, I don't want you to think of it as blue, though, because that's not really true. Now, coloring these vessels red and blue are very beneficial, though, okay? So we will be thinking about the blue vessels as deoxygenated blood and the red vessels as oxygenated, as long as you realize the blood's not really blue, okay? All right, so everything that's blue here is showing deoxygenated blood. So let's follow that first. Notice here called the superior vena cava, right here. This blood is coming, the blood's been in the brain. The brain has used all the oxygen that it needs. It's bringing it back down to the heart. It's coming to the right atrium, the upper part of the right side of the heart. There's a pump, right? The muscle contracts. It squeezes it out of the right atrium down into the right ventricle. And then the squeeze sends it up through the semilunar valves into the pulmonary trunk, the left and right. We have two lungs, one to the left, one to the right, right? There's the right pulmonary arteries. Going to the lung. They go to the lung, they get oxygenated, they come back to the heart. Here we go. Into the left and the right pulmonary veins. Notice they're red though, even though they're veins, because it's coming to the heart, right? And those veins will come into the top part of the right side of the heart. Okay, that's the, left side. The left atrium gets that fresh oxygenated blood. The pump sends it down into the left ventricle, and that giant pump sends it up through the aorta to the head and to the rest of the body. Okay, so that's the cardiac cycle right there. Here are those valves. Here's our mitral valve in our left atrium. Right here, it's very large. And they're tricuspid. See, it's got three parts. 1, 2, 3. That's the tricuspid. And here's our valves over here, our semilunar valves over here. All right. So our cardiac cycle, blood is pumped to the heart via electrical commands. What's causing it to pump? Electricity. Okay, fancy electricity. Both ventricles and both atria contract simultaneous with each other, but the atria are just before. That's why it's bump, 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 bump. Okay? Now, we have these things called nodes. These are the electrical signals that are inside the, well, one of them's in the atria, right? So, and the other one's in the septum, that part in the middle right there. So the sinoatrial node sends a signal to the atria, and the atrioventricular node is the AV node. So the SA node and the AV node send signals to the ventricles. That AV node is in the septum area, I think. So step 1 is called early diastole. The heart is relaxed, the ventricle is filling, okay? Then we have systole, the atria contract. We'll watch a video to help us here in just a minute. Then we have ventricular contraction, boom, all right? Ventricular ejection, it's gonna send the blood out either to the lungs on the right side or to the body on the left side. And then we have ventricular relaxation, the atria begin filling again. So I want you to think for a second, when you flex your muscle in your arm, does it make a noise? No. Right? Doesn't make a noise. So why, when you listen to your heart, which we will do next time, is it going thump, 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 thump? Well, it's not the muscle. The muscle contracting is what's causing the blood to move, but what's causing the sound is the slapping shut of those valves. Okay, so it pushes through, then slap, that valve closes. And then the other side closes, right? So pump, pump. So it's slap, 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 slap. Those are valves closing. So that's what you hear on that, okay? All right, so here it is. We can see our AV node. Our SA node is way up here, and our AV node is right here. It's the one near the septum. The septum. See the septum is that tissue in between the two, right? And you can see those vessels that are traveling down through here, okay? And that's going to excite that muscle. We know that muscular tissue is excitable. All right, and that's what's going to cause it. Boom, boom, right? So we're going to cause those contraction, contraction. And then here's the valve and the valve. Slap, slap. Okay. I had a student, a very good friend of my family, who ended up getting born with an extra set of electrical tissue inside here, okay? It was called, at first they were calling it Wolff-Parkinson-White syndrome, and then they changed it a little bit. Anyway, it caused him to have his heart rhythm went into, I'm going to call it a short circuit. So instead of going, contraction, contraction, it actually started making a circle right here. So a short, and his heart would just quiver. So when he was born, he had a heart rate of about 300, and that was hard to count. So he got put into the NICU unit with all, he was like a 9-pound baby, and he's in the NICU unit with all these 1-pound babies because his heart was beating so fast. And so he had to have a monitor for the beginning of his life so that he would know when he went into this little rhythm, this arrhythmia, causing what is known as tachycardia, meaning fast heart rate. And it was very interesting. I'll tell you about it next week where I can explain it a little better. Is that when we call it an episode, he would go into an episode when all of a sudden this was beating, 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 causing his heart rate to go really fast. So this Holter monitor he was wearing would let us know when he went into this deal. And we had to try to get him out of it. Now, I'm going to tell you, this is really bizarre. I don't know if they do this anymore. This kid is now probably 25 years old now. But what they had to do when they would get him into, he would get into this arrhythmia is literally, y'all, first of all, they would hold him upside down and see if that would help. And if that helped, that was good. Something about that pressure of his head being down caused that to happen. If it didn't work, they literally, I kid you not, had to dunk his head into a bucket of cold water. Yeah, and it caused that kind of that shock or whatever to snap out of it and cause his heart to start beating regularly again. Okay, as he got older, he knew when he was going into an episode because he could feel it. And he would say, hey, Mom, I'm in this episode. So the first thing they would do to him is he'd go stand on his head. And when he went to go stand on his head, he would wait for it to get, better, this kind of thing. As he got a little older, they actually were able to put him on a medication that was able to control it. And then when he got to be, I think he was 12 or 13, when his heart was big enough, they literally went in to his heart with a little radio, like probe, some sort of deal. And they went in here and burned out those extra little electrical bundles that were in there. And it fixed it. Now he's perfectly fine. He has no trouble with his heart at all. He played football in high school and that kind of thing, but a very interesting condition that he had. All right. Here is a really good picture of the flow, the way it works. I think you're supposed to draw this in one of your boxes. Okay, so as long as you know it goes from right atrium to right ventricle to the lungs, left atrium, left ventricle to the aorta and body right there. Okay. We're going to watch our cardiac cycle video right now.
Ventricular contraction
Sorry
causes
about
the
that quality.
atrioventricular valves to close, which signals the beginning of ventricular systole. The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open. When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole. Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.
Okay, see how that works. All right, let's talk about our blood vessels for a little bit. They're actually different. We can see our artery right here. It's actually got a much thicker, smooth muscle right here than the vein. The vein, the smooth muscle, is thinner right there. Also, veins have valves in them. So the vein and arteries are composed of three tunics: the tunic intima, the tunic media, and the tunic adventitia. Adventitia. The tunics and veins are much thinner, allowing for more blood volume. Capillaries don't have any tunics at all. They're very, very thin. They allow for very easy gas exchange. Very thin tissue right here for our blood vessels to exchange that gas. And our arterioles still have tunics, and venules have smooth muscle. So we see vein, venule, and capillary. Artery, arteriole, capillary. Okay? Now, think about why the smooth muscle here is thicker right here, because remember, if it's pumping, the heart is pumping it through the aortas away from the heart. It has to get places, right? This is why you can feel a pulse. If you put your fingers on your wrist or on your neck, you can feel that pulse. That's an artery you're feeling right there, because it's getting that pump from the heart, really that big heavy-duty pumping. Whereas the valve is really passively just being pushed. The blood is being pushed along back toward the heart because there's blood in front of it that's pushing it, okay? So it goes from the artery into the arteriole, into the tissues right here. So this is the capillaries, which are going to be bathing the tissue around it with the oxygen and picking up the carbon dioxide. Then it goes into the venule and up to the vein and back to the heart, okay? All right, what I think that's pretty cool about vein, they have valves in them. Remember, it's just being pushed up passively, sort of, from you don't feel the pulse in these. But because it has that valve right there, it won't allow for backflow of the blood. Okay, that's why they have those. That backflow will be stopped as it's being pushed along. Okay, what about blood pressure? It's the pressure exerted by the heart on the vessels, those blood vessels, namely the aorta and the arteries. And the increased pressure can cause damage to the vessels. That's called hypertension or high blood pressure. Blood pressure is determined by measuring the maximum pressure called the systolic. That's the heart contracting. We'll watch a video in a minute. And the minimum pressure called the diastolic, or the relaxing time, where the diastole is measured. Normal blood pressure should be about 90/60. That's pretty good, actually. It can be a little higher than that. High blood pressure, I believe they're starting to change their mind about this. It might actually be lower than that now, considered high blood pressure. If you think about it, if I've got a lot of pressure slamming against my vessels, you can see how that might cause damage over time, right? So, because it's just a lot of pressure there. And so what could cause this kind of thing? We'll look at the video here in just a minute. This is how you take it, all right? So what you do is you take your arm, which is really close, in level to your heart right here, and you squeeze it so that no blood is getting through. It's not very comfortable for a second right there. And then as you start to open it up, you start to hear the sound here. That's the systole right there, the arteries opening and closing, and then we get to the diastole right here, and then no more sounds because the artery's fully open right there, okay? I have two different, this is called a sphygmomanometer, something like that, and I have two different ones. I have an automatic one that's run by batteries. I hope it's still working. And then I have the one you use with the stethoscope in your ears. I find it difficult to do. Doctors and nurses have to do it so often that it's very easy for them. In fact, it's probably easier than putting them up to one of those machines or something. But we will test your blood pressure next week. Let's watch a little video about it.
If you lined up all the blood vessels in your body, they'd be 95,000 kilometers long. And every day, they carry the equivalent of over 7,500 liters of blood. Though that's actually the same 4 or 5 liters recycled over and over, delivering oxygen and precious nutrients like glucose and amino acids to the body's tissues. All that blood exerts a force on the muscular walls of the blood vessels. That force is called blood pressure, and it rises and falls with the phases of the heartbeat. It's highest during systole, when the heart contracts to force blood through the arteries. This is your systolic blood pressure. When the heart is at rest between beats, blood pressure falls to its lowest value, the diastolic pressure. A typical healthy individual produces a systolic pressure between 90 and 120 millimeters of mercury, and diastolic pressure between 60 and 80. Taken together, a normal reading is a bit less than 120/80. The blood traverses the landscape of the body through the pipes of the circulatory system. In any plumbing system, several things can increase the force on the walls of the pipes: the properties of the fluid, extra fluid, or narrower pipes. So if the blood thickens, a higher pressure is needed to push it, so the heart will pump harder. A high salt diet will lead to a similar result. The salt promotes water retention, and the extra fluid increases the blood volume and blood pressure. And stress, like the fight or flight response, releases hormones like epinephrine and norepinephrine that constrict key vessels, increasing the resistance to flow and raising the pressure upstream. Blood vessels can usually handle these fluctuations easily. Elastic fibers embedded in their walls make them resilient. But if your blood pressure regularly rises above about 140/90, what we call hypertension, and stays there, it can cause serious problems. That's because the extra strain on the arterial wall can produce small tears. When the injured tissue swells up, substances that respond to the inflammation, like white blood cells, collect around the tears. Fat and cholesterol floating in the blood latch on too, eventually building up to form a plaque that stiffens and thickens the inner arterial wall. This condition is called atherosclerosis, and it can have dangerous consequences. If the plaque ruptures, a blood clot forms on top of the tear, clogging the already narrowed pipe. If the clot is big enough, it can completely block the flow of oxygen and nutrients to cells downstream. In vessels that feed the heart, that will cause a heart attack when oxygen-deprived cardiac muscle cells start to die. If the clot cuts off blood flow to the brain, it causes a stroke. Dangerously clogged blood vessels can be widened by a procedure called an angioplasty. There, doctors thread a wire through the vessel to the obstructed site, and then pass a deflated balloon catheter over the wire. When the balloon is inflated, it forces the passageway open again. Sometimes a rigid tube called a stent is placed in the vessel to help hold it open, letting the blood flow freely to replenish the oxygen-starved cells downstream. Staying flexible under pressure is a tough job for arteries. The fluid they pump is composed of substances that can get sticky and clog them. And your typical healthy heart beats about 70 times a minute, and at least 2.5 billion times during an average lifetime. That may sound like an insurmountable amount of pressure, but don't worry. Your arteries are well suited for the challenge.
All right. There we go. I think it's going to play it again. Yeah, so I'm finished now, y'all. That is it for this module. We made it through it. Sorry it went a little bit long. But next week when we get together, we'll have a quiz, and we will have a really cool lab. All right. Wonderful. Have a great week. Stay safe. Bye. See you next week.